Hello everyone, and welcome to this brief introduction to Stata. My name is Todd Smith, and I'm going to be walking you through this session. Now, the intent tonight is not to make you an expert. It's not really even to make you proficient in data analysis using Stata. The intent is to get you over the initial trepidation with using a new software. Now, I'll try to go slowly, but if I do go too fast, you can always pause the recording or run it back. Just use it at your own speed. So the agenda tonight is first I'm going to go over the basic Stata user interface. Next, I'll show you how to get some data into Stata, which is always the first step. Then I'll move on to talking about a little bit about the structure of data and types of variables that Stata uses. And then finally, I'll show you a few of the basic Stata functions, the things that we always do at the beginning of any data analysis project. So moving on to the user interface. When you first open up Stata, this is what you see. This basic user interface is the same regardless of whether you're using a Windows environment or a Mac environment. There are four windows. The first window is the command window. Now, the command window is where you tell Stata what you want it to do. There are three basic ways to get commands into Stata. One is to type the command directly into the command window. In order to do that, however, you need to know the command name and the correct syntax. Alternatively, you can use the drop-down menus from the menu bar. I'll show you more about this in a few minutes. The third way is from a do file, which is what Stata calls programs, which is just a list of commands that Stata will perform in the succession that they're written in the program. Next is the results window. Now, this is the window where you'll see the results from the operations that you ask Stata to perform by giving it the commands in the command window. The third is the variables window. Here you'll see a list of the variable names and the metadata of the variables of the data set that you currently have in the local memory and that Stata is using at the time. Right now I have no data in the program, so there's no metadata shown. The top window shows the variable name and the variable label. Now the difference is the variable name is used by Stata when performing operations from that data from that variable. This has certain restrictions about what variables can be named. For example, variable names cannot include spaces or punctuation. They can include numbers, but they cannot begin with a number. And this may come up later, but variable names are case sensitive. In the bottom, the variable label, however, is more user friendly, more human friendly. Might be a more descriptive title for the variable of that variable. Now, on the bottom, you'll see other metadata about the variable, what type it is, the format that it's using. You can add notes, you can add labels to not just the variable, but the values that are in that label. We'll talk more about that in a few minutes. Finally is the review window. Now this is just a running ticker tape of the commands that you have told Stata to do in the order that you've told them to do it. You should note that you can access previous commands directly from the review window, or you can access variables directly from the variable window and import those directly into the command window, which is a handy time-saving tip. I'll show you more about that in a bit. Other parts of the user interface are the data browser. You can click on this button to actually see the data that you currently have in the local memory. But caveat, do not ever use the data editor button. This will allow you to directly alter the data that you currently have in memory in much the same way that you can in an Excel spreadsheet, but this is really not a good practice. If we ever need to alter the data, we should do it in a way that we will have a record of the changes that we've made. And finally, if you click on this button, you can open the do file editor, which is really just a fancy specialized text editor where you can write programs for Stata. We'll talk a little bit about this later, but we're not going to get into this tonight. Okay, we're going to move into actually using Stata now. And the first operation we're going to do is we're going to get some data into Stata. And there are three basic ways to do this. Now the first and the easiest is simply to open a data set that was already saved in a Stata data format. Now these types of files you can recognize because they'll have a .dta file extension. And like most other programs, for example, Word or Excel, you can open these types of files from directly within Stata or simply by double-clicking a .dta file from your Mac Finder or your Windows My Computer Explorer window. Now your computer should recognize that this is a Stata file and will open it in Stata. So to give you an example, here I am, I have Stata open. I can click File, Open, navigate to my EMPL 2014 file. Now I already have stored these data sets under Datasets. And what we're going to be using is the resid.dta file. Now this is the file that you'll be using for your initial homework. I highlight that and simply click Open. And you'll see that the variables populate here in the variables window. The results repeats the command back to me that I ran, the use command, which is what we use to open Stata data files. And you'll see in the command window that this is the first command that I ran. Now say I wanted to clear this data out. The way to do that, go to the command window and simply type clear and press enter. And you see that it clears out the data that is in memory. And then there are no more variables in the variable window. It repeats the command back to me, and it's ready for the next command. Now, like I told you, you can go to the review window, simply click on the command that you would like to do again, in this case, the use command. We're going to reopen this data file. Press enter, and again, it repopulates the variables, and I have repeats the command back to me. Now, the other way to do this, let's clear this data out again. The other way to do this is to open it from the Finder window. I can navigate to my EMPL 2014 file, click on Datasets where that dataset is open, and click on the .dta file. Now, if I double click on that, like I said, it will open directly in Stata. Now, the second way to get data into Stata is to import data that's created in another program and stored in a tabular format. And by that, I just mean stored like a table. Now, this is most commonly data that might be created in Excel or stored as a delimited text file. And by delimited, we simply mean that each line of the text in the file is a separate row for the table, and each column in that line of text is delimited by a specified character, generally a comma, in which case we refer to this as a comma-separated file or a .csv, or a tab, in which case we refer to it as a tab-separated file or a .tab. So to do this in Stata, first let's clear out the data that we have. Again, go to the File menu, go all the way down to the Import option, and it opens this other list of options. Let's select Excel spreadsheet. And now we can browse directly to that. And again, I'm in the correct folder. I can click on the resid.xlsx file, click open. And it's nice because it gives me a preview of what's in the data file and the variable names and what the data looks like. So I can see if it's going to import correctly. Now this is almost correctly, except that these in the first row are the variable names. They're not the first row of data. So I want to select this option to import the first row as variable names, and now you see that they'll import as variable names. You can select OK, and it imports the data just as if I opened it as a DTA file. It populates the variable names. You can see that it returns the command, which is the import command, import Excel, and then the name of the file name, and then these few options. We'll talk about options in a bit later. And it's as easy as that. Now, we're going to talk about one other way to import a .csv. So let's clear out this data. And I've already created this resid data. Go back to import. But this time, instead of selecting Excel spreadsheet, I'm going to select text data in a delimited format, .csv. So I'm going to select text data. Again, just like before, browse to the file name. It recognizes that it's a CSV, or that CSV is one option. I can select that, click on Open, and again, it gives me a preview. Now this time, use first row for variable names. It's selected automatic. Now, I can say always, I can say never. Let's just leave it as automatic because that looks like that's going to import correctly. So then I can select OK. And again, I have the variables imported. It imported 18 variables, 1,445 observations. It tells me what the command is, so I can know for later reference. Okay, so that's it for getting data into Stata. Like I said, the third way, there is a third way, and that's to use an input command. And this we might do in a do file. It's really not that useful for large data sets. So I'm not going to talk about that anymore today. What we're going to do, though, is take a closer look at the data structure that's used by Stata. Now, each row in the data set is a separate observation. Now, observations can be whatever we want them to be, but we need to be clear about what they are. For this particular data set, if we look at the data dictionary, the code book, we can see that the RECID data consists of records of 1,445 convicted individuals released from North Carolina prisons during the period July 1st, 1977 through January 30th, 1978. So this particular data set, each row is a separate person, a separate convicted felon released from North Carolina prisons, so an individual. Alternatively, if we were to have data at a particular time for population of countries, then each observation might be a country, in which case each row would be a separate country. We could also have data for a single country across a number of years for population, in which case each row would be a separate year. Not a separate country, but a separate year. We could also have data on multiple countries for multiple years, in which case each observation is a country year. I'll show you an example of that in a minute. Now, in our data set, each column represents a variable. And in each cell is a particular data point for individual one about the variable drugs. So now I'm going to switch data sets. So let me close this. Let me clear out this data. And I'm going to open the data set that I created earlier. It has data from the World Bank about countries, population, and GDP. Double click on that. You'll see it populates with a new set of variable names. Now if I open the data browser, you'll see that this data looks quite a bit different than the other data. I have this red and blue and black. Now each of these represents different types of data. Basically, Stata recognizes two data types, numbers and strings. Now numbers are exactly that. And whether they are integers, double precision, floating point numbers is just about how they are saved and how much memory they take up. Essentially, they are treated the same way by Stata. Sometimes the numbers might be identifiers, in which case the quantity, the value, is meaningless. It's just a unique identifier, such as a social security number. But Stata doesn't know this and will treat them like any other number, so we have to be careful about that. Numbers are displayed in the data browser as black. So you'll see this year variable is black. That Stata recognizes that as a number. Population recognizes as a number, and you can see that they're very large numbers. GDP, also very large numbers. Now this is important because Stata recognizes or treats missing variables as a dot. You'll see these dots indicate that the data is missing for these observations. Now the observations in this case are different individual countries in individual years. So this is Aruba in 1960. This is Afghanistan in 1960. If I scroll down, you see that I go from 1960 to 1961, and I have Aruba in 1961, Afghanistan in 1961, and so on. The second type of variable, I recognize that this is Aruba because it says so. Now this is red. This means that it's a string variable. And that just means a string of characters. Now Stata can't do any type of analysis with strings. These are generally just useful to us humans because they might have information that is not easily quantifiable. Now sometimes it is useful to have variables be numbers so that Stata can use them for analysis, but for us to know what the variables mean if the number doesn't have some inherent meaning, like 1960, 1961, or a population number. Now for this is especially useful for categorical variables, and in this case we can assign labels to the values, and that's what these blue variables are. So you see this variable is actually a 4. Stata recognizes it as a 4, which I can see here, but I've previously assigned a 4 to mean lower middle income country. Now this is actually a 1, but I've previously assigned 1 to mean East Asia and the Pacific. A low income category is represented by a 3. High income non-OECD countries are represented by a 2. Sub-Saharan countries are represented in the region variable by a 7, and so on and so forth. Later we'll get into more about why this is useful to assign variable names and how we can do that, but that's for a future session. What we're going to do right now is go back to the resid data and talk about some basic Stata commands. So let's close the data browser. Let's clear out this data. Let's go back to the resid data, and remember I can do this simply by recalling the command that we used earlier. So I can select that, go down here, press enter, and now I have the resid data back in. Let's open up the data browser and take a look at this. And you can see this is just a series of zeros and ones. So these first variables, black, alcohol, drugs, super, married, felon, these are what we refer to as dummy variables or indicator variables. Now if I don't know what these are coded as or what these zeros and ones mean, I can find out by referring to the data dictionary or the code book. So I can see that the variable labeled black means that that's individual. If it's a 1, that individual is black. For alcohol, if it's a 1, that individual has alcohol problems. Now the easy way to remember this, and the way that dummy variables should be coded, is to imagine that there's a question mark after the variable name. Alcohol, question mark. If yes, then it should be a 1. If no, it should be a 0. 1 means yes, 0 means no. Drug, is there a history of drugs? Drug, question mark. 1 means yes, 0 means no. So I can go back and I can see that this individual 1, does have an alcohol problem, does not have a history of drugs. So let's close out of this and talk about how we can get some summary statistics or find out a little bit more about each of these variables. So the first, the most basic summary statistic for any variable is the mean. So how do I find out what the mean of black is, what the mean of alcohol is, what the mean of drugs is? To do that, I don't know what the command. If I don't know what the command is, I can go to statistics, summaries, tables, and tests. So select this first thing in the list, summary and descriptive statistics. Means, that looks promising. Let's click on means. Variables, I have this drop-down list. I can select black and see what is the mean of black. I can click on OK, and that will give me the mean. The mean is 0.485 for black. Now remember that means, if it's a 0 or a 1, the mean is the proportion of the is really interpreted as the proportion of ones. So this means that 48.5% of my observations for that variable are a 1, or 48.5% of the observations in this data set, the individuals in this data set are black. That's useful, but I want more statistics about that. So let's go to statistics, summaries and tables, summary and descriptive statistics. Let's choose this, summary statistics. Now this is the most useful. And so let's look at a different variable. Let's look at alcohol problems. Now I can, just as before, I can select OK and it will submit the command and close this dialog box. If I don't want to close this dialog box, however, I can just select the submit command and it submits the command, but it does not close the dialog box. And here I can see that summarize alcohol was the command that it submitted. And it gives me the mean and the standard deviation. It gives me the minimum and the maximum. Now this is useful because I can see that this is an indicator variable or a dummy variable. The number of observations is 1,445. And 20.96% of the observations in this variable are ones, or 20.96% of the individuals in this data set have alcohol problems. Now I can go back to my summary command, and I don't have to do this for just one variable at a time. I can select multiple variables, drugs, super, married, and select OK. And it will give me the summary statistics for each of those variables. Now this is the first, which again are the means, the standard deviations, the minimum, and the maximum. And I can see that these are all indicator variables. Now summarize is really the first command that you should memorize because it's a little bit cumbersome to have to go through the dialog box every time when there's a much shorter way to do this, and that is just to type sum. Sum is short for summarize. So I can just type sum, and then I can list the variables that I want to have the summary statistics on. Now, I can type them in individually. I can type black, or I can select them from my variable list. So if I click here, it will import alcohol into my list of variables that I'm going to get the summary statistics on. So let's get drugs, super, married. Now let's get some of the ones that I know are not indicator variables. So education, I know because I've already looked into it, is not an indicator variable, and age is not an indicator variable. So go back here, hit enter, and now I have the summary statistics on all of those variables. So just like I said, education and age are not indicator variables. The minimum is 1 for education and the maximum is 19. The minimum is 198 for age, the maximum is 933. And these are the means and the standard deviations, so I get a sense of the spread as well as where the central tendency is. And next, we're going to talk about the tabulate command to generate tabulations. Now, there are two types of tabulations, one-way tabulations and two-way tabulations, also known as crosstabs. So to get to the tabulate or tab command, go to statistics, summaries, tables, and tests, and frequency tables. So first, let's create a one-way table. And here we're going to select the categorical variable that we want to tabulate. Let's take a look at married. Submit. Now this just gives me the frequency of observations in the married, for the married variable that are 0 and 1. So the frequency of zeros is 1,076 zeros versus 369 ones. Now this is the percentage of ones, and this is the percentage of zeros. Now let me cancel out of this. Now remember I said that the mean for categorical for dummy variables is simply the proportion of ones. So here I can see that the mean is 0.255, and here it's telling me that the percentage of ones is 25.54. It's the same number. So just proves what I was saying. More useful, however, is a crosstab So to do that, we go back to statistics, summaries and tables, frequencies and tables. Let's go to two-way tables. So I can look at the row variable. This time, let's look at married. And the column variable, let's look at black. Okay. So now I can see how many people are married and black, or how many people are not married and not black, or how many people are married and not black, or not married and black. So if it's a 0, 0 in this corner, those are people that are not married and not black, 541. People that are married and black in this lower corner is 166. So this can be a very useful command. Now the shortcut for this, which you should probably also know, is tab, is short for tabulate. So I can do the same thing with married by typing married and black, or I could select them from the list over here. Hit enter, I get the same thing. Now there are useful ways. Now this doesn't necessarily show me the percentages, but I can get these percentages using options. Options are simply small variations in the command that does something slightly different. So if I don't know what the syntax or the command is, again I can go to statistics, summaries, tables, and tests, frequency tables, two-way tables with measures of association, and married selected here, and black. You can see I've already selected here within column relative frequencies. So this is the option that I want to select, and it's going to give me the column percentages. So I click OK, and now I have, again, the frequency counts for 0, 0, for 0, 1, for 1, 0, and for 1, 1. But this time it's telling me the within column percentages. And you see column percentage is this is the frequency and column percentage. This is my key. So 72.72% of the zeros in the black variable are also married. Are also zeros for married, sorry. And if it's a 0 in black, 27.28% are 1 for married. So these both of these numbers add up to 100 in this column, and both of these numbers here add up to 100 in this column. Now to do that, you can see the command here was tabulate married black, comma, column. So this is the option. Options always go after the comma. So what do you think the option, if I want to see what the within row frequencies are? Tab, select married, black. I'm going to type comma, row. If you didn't guess that, that's what it is. So now I have the row frequencies. So you can see now that they add up to 100 across the rows. So of the zeros for married, 50.28% of them are also 0 on black, and 49.72% of them are 1s. Of the married ones, 55.01% are zeros, and 44.9% are 1s. So it can get even more complicated, and it might get a little bit confusing. But if I wanted to know the row frequencies and the column frequencies, I can do that. So I can select this last command that I did, tab, married, black, row, and I can add on column, or for short, just COL. And I can enter that. And now you see the first number in the cell is giving me the frequency, the second is the row percentage, and the third is the column percentage. So these two numbers add up to 100, and this number adds up to 100. So that's the tabulate command. It can be very useful for exploring relationships between two categorical or indicator variables. The last thing that I want to talk about tonight is the correlate command. Now, the correlate command gives us the correlation coefficient between two variables, which is just a measure of how closely correlated the variables are. So we could do this with the same with married and black to see if there's any correlation between these numbers. See if these these tabulations mean anything. So to do that, if I don't know what the syntax is, statistics, summaries, tables, and tests, summary and descriptive statistics, correlations, and covariances. Halfway down this list here. So I just have to select the variables that I want to see the correlation for. Black, married. I have to select at least 2 in this case. Submit. And it tells me, let's cancel out of this, it tells me what the correlation is. It's a negatively correlated 0.0413. Now, I'm not going to get into what this means. We'll get into the later in the class. I just wanted to introduce the command because you'll be using it very shortly. The shorthand for this is CORR, C-O-R-R. And again, I can select the variables that I want to see the correlation on. So maybe I want to see if there's a correlation between people who are married and people who have a history of drugs. And it's a negative correlation and not much of a correlation at all. I can look at more than just 2 variables at a time, but it's going to show me the correlation between any 2 variables. So let's see CORR. Let's just select black, alcohol, drugs, married, and education. And now, see, the way we read this table now, this correlation matrix is what we call this. Correlation matrix between alcohol and black is negative 0.119. Between drugs and alcohol is negative 0.0087. Between black and education is positive correlation of 0.1064. So that's the correlate command. And like I said, Professor Wilson and Professor Stolp will get into more about what these correlations mean in class. And that's the last thing that I wanted to show you. So I hope you've learned how to get data into Stata. You should have learned some about the data structure that Stata uses and different types of variables in Stata. And finally, we learned about the summarize command, about the tabulate command, and about the correlate command. And I hope it was useful. So if you have any questions, email me or come see me or Skype me during office hours. So that's the end.